how in the world did you decide to go looking in the ocean for new compounds?
I have to say my day job is really a dream job. I love sports, and drug discovery is really a team sport. I have the privilege of interacting, working with chemists, biologists, synthetic biologists, and pharmacologists to form a team that has the same vision of developing new therapies from marine environment and bring these compounds, these discoveries, really from the beach to bedside.
I like that, beach to bedside. And we were talking off camera, so you are a scuba diver, and you've trained and been in Hawaii, and you've been at Scripps Institute in Southern California, and now you're here in Florida. I've noticed a pattern that you seem to like warm water. And those of us who live in Southern California, or California in general, somehow we don't have warm water.
I know. And we didn't have warm water in Germany as well.
No.
So, as you know, you can tell by my accent. So I grew up in Germany, landlocked, but I read in the literature about amazing discoveries that were made at the University of Hawaii, which is the birthplace of marine natural products chemistry. So the pioneers really of the field worked at the University of Hawaii. And I had the privilege of working with the first professor who ever looked at marine cyanobacteria, a type of also called blue-green algae, as a source of pharmaceuticals. So I saw the amazing structures and the amazing activity, and I wanted to be part of that. I thought that's so fascinating, and I applied, got into the university, and traveled around the world from Germany to Hawaii to train with the best, to train with the pioneers, and had to get scuba certified, right? So that's where my, adventure started, adventure in the ocean. And yeah, our starting point, as you mentioned, is the water. We start by snorkeling or diving. It's really a treasure hunt in a wetsuit, diving for chemical gold.
Well, I think many people realize that so many medicinal compounds, pharmaceutical agents, have their basis in plants, and we naturally kind of think of terrestrial plants, but ocean plants predate anything that's happened here on land. And you mentioned cyanobacteria. That's a fancy word. Most people have heard of spirulina, a blue-green algae, and that's a type of that algae that you mentioned.
That's correct.
So take our listeners, why get interested in the sea? Because the sea came first, and maybe there's some pretty cool stuff happening down there.
As you mentioned, most of the drugs actually on the market are based on natural products, either natural products themselves, but most are at least inspired by terrestrial natural products, products from plants, soil bacteria, fungi. But if you look at our planet, 70% of the planet is covered by ocean. In the three-dimensional space, it's 95%. And the greatest biodiversity is in the ocean, not on land. So can you imagine, can you extrapolate, how many discoveries are just waiting for us to be developed into potential therapies? But then again, there are also so many organisms. Which organisms to focus on? Life presumably started in the ocean. So obviously, organisms there had long evolution times. But you really want to work with the best, right? Always. You want to work with winners. Cyanobacteria, to me, are the winners. They're the first organisms on the planet, the oldest organisms on the planet. They had 3.5 billion years of evolution. So they're now a thing or two about aging well and making it in this environment, planet Earth. So we really want to learn from those cyanobacteria and get inspired by the chemistry they are producing.
Yeah, Hendrik is very modest. He's actually made discoveries that have been the blueprint for FDA-approved anti-cancer drugs, like Dolastatin 10. So this guy's impressive, folks. What does that say about the ability of the ocean to really be a fundamental place to look for drug discovery pipeline?
So far, there's over a dozen, 15 or so marine drugs on the market. And 1/3 of them, as you pointed out, are based on dolastatin 10, which is a cyanobacterial compound that we discovered in Florida, as well as in the Pacific Ocean. But this is really just the tip of the iceberg. So in every collection that we look at, we find completely new compounds. And these new compounds occupy new chemical space. That means they probably translates into completely novel pharmacology. And we just, looked at less than 1%. Can you imagine if you have the ability to access the remainder of the ocean, how many potential therapies we could find to address unmet clinical need?
And this is something I've been interested in for many years. Why do plants and algae make these compounds in the first place? Are they survivability factors that protect themselves from a harsh environment? Or, they don't just make them for fun.
Yeah, so I asked them that question. Yeah, no, it's a very good question. Of course, we can only speculate, but especially the ocean is a very competitive environment. It's a battlefield there, battlefield for nutrients and for space, sometimes scarce nutrients, right? And the organisms we are interested in, they're sessile. They don't have teeth to defend themselves. So what do? They produce chemical weapons. Chemistry to defend themselves against predators, competitors, but then also really use this chemistry as a language to communicate. So the more communication is going on, that means the more chemistry. And that's why we really focus on areas of greatest biodiversity, like coral reefs, where you have a large number of different organisms, because there's a high level of communication and defense. So we are doing targeted collections in those areas of high biodiversity, which gives us ultimately a rich arsenal of weapons that we are trying to repurpose essentially for biomedical applications.
Now, you've also done other major cancer drug discovery. You discovered gator-bulin from cyanobacteria. Now, since we're talking gator-bulin, I've got to ask, University of Florida Gators, was that a name of coincidence or something you were having fun with?
We're always having fun. I think that's the recipe for success too, that it's not just a job, right? It's really passion that happens to be a job. But I also view it as, an obligation to society to really unlock the therapeutic potential of these natural products. Because we are able to do it, we must pursue it. And yes, gator-bulin, it's my favorite name. One of the beautiful aspects of our work is all these compounds are new. So we can name these compounds because nobody has ever seen them before. And we try to make it catchy, but also descriptive so that people know where the compound comes from and what it potentially does. It targets tubulin, so it incorporates the name bulin, tubulin for the molecular target, and the name gator, of course, really gives credit and tribute to the discovery at the University of Florida, to the team that I led. And it was a long journey to really characterize this molecule in detail. It started with the cyanobacteria bloom right here nearby in Fort Lauderdale in 2005 and 2006. This was where the original collection happened. And we knew there's something completely novel in there. And it took a decade of development, early stage development, to understand the chemical makeup, to reproduce a compound in our lab and at the University of Florida, to identify the molecular target and the mechanism of action so that we actually have biomarkers for preclinical studies and hopefully in the future clinical studies. So it's really a tribute to the team at UF, the Gators, and also, our global collaborators, international collaborators in Europe as well, which we call now the Global Gators, part of the family.
But we probably shouldn't expect to see gator-bulin in Gatorade anytime soon.
Not in Gatorade, but hopefully eventually on the market in one form or the other.
Just don't want people to go to the store looking for that ingredient in Gatorade. So you mentioned this is a very long process. This is like there's an algae bloom here in Fort Lauderdale, and you say, oh my gosh, look at this, there's a compound here. But it took 10 years to get from this algae bloom to actually having something with value for human clinical trials.
And we are not there yet. The journey has still just started. And sometimes these discoveries can take a few months only, and sometimes it takes 10 years. It really depends on the novelty of the discovery. And the more novel, the harder to predict, the path it will take in order to fully understand and characterize a molecule. In this case, we stumbled over, novelty at the level of structure, at the level of chemical synthesis and of how to make it, and at the molecular and atomic level, how this compound interacts with molecular targets. So many elements of novelty delay the process, but make it even more rewarding if we are successful at the end.
So I think a lot of our listeners wonder, okay, so what's the difference between the drug development pathway that needs FDA approval that you and I know take many years of clinical trials versus taking a similar discovery and turning it into a dietary supplement?
Our focus for the most part has been on the discovery of therapeutics, and that's where we use these cyanobacteria as a starting point to really address diseases with unmet clinical need. It's a long road to begin with. You have to reproduce the molecule in the lab. You have to identify the molecular target. And you have to develop a data package that allows you to register the compound with the FDA and get IND status, investigation and new drug status, and which then allows you to enter clinical trials and perform safety studies in phase 1, as well as efficacy studies in phase 2. So that's an extremely long process, as well as, expensive process. But we are really not starting with a dietary source, and that's why these, studies are, of course, have to be rigorous and are very important. You really have to start with a dietary source that has proven to be safe already and can be taken in a safe manner. And for us, it's important that we show efficacy of these potentially dietary supplements. And our focus in that space has been on seaweed, because it's a proven dietary source that has been ingested in many, particularly Asian countries, and has been inversely correlated with the incidence of cancers, particularly in Okinawa, where the seaweed consumption is the highest, and also has been shown that, Okinawans have a long life expectancy, and there's a potential link to seaweed consumption. So that's why we entered the area of seaweed,